Hello, grade 12 students. Here we start with chapter 6, the role and the components of the immune system. To start with, blood is divided into main parts. We have the plasma, which is serum, and the cells. Plasma is about 55%. It contains the water, salt, sugar, and the antibody. Cells, which compose about 45%. They are composed of three main components: erythrocytes, the red blood cells. The job of these is to carry the oxygen and carbon dioxide; platelets, which play a role in blood clotting; and the leukocyte or blood cells. They play a role in immunity. These leukocytes, they are divided into myeloid and lymphoid. Let's talk about myeloid. Myeloid, there are three components: granulocytes, and monocytes, and the mast cell. Granulocytes, the main characteristic is having a multi-lobed nucleus. They are neutrophils, eosinophils, and basophils according to the type of the granules. Monocytes, they have horseshoe-shaped nucleus. They become mature and they engulf microbes by a process called phagocytosis. Mast cells, they have round nucleus and they secrete histamine and play a role in allergy. Lymphoid, we have two main parts: B lymphocytes and T lymphocytes. B lymphocytes, they mature into plasmocytes or plasma cells that secrete antibodies to kill and neutralize the antigens. And we have T lymphocytes that are composed of T4 helper and T8 killer. T4 helper secrete cytokines, and T8 killer secretes perforin and granzymes to kill the infected cells. Okay, we want to talk about the HLA. Let's talk about the four types of graft. We have autograft, isograft, allo, and xenograft. Autograft, it's a graft that's done within the same individual, while isograft is done between two identical twins. Allograft, two genetically different individuals belonging to the same species, while xenograft, two genetically different individuals belonging to different species. Blood groups, another self-marker. Agglutination. What is agglutination? Agglutination is the binding between an antigen and its corresponding antibody. It's called agglutination. So let's talk about blood group A. Blood group A, it has the antigen A. So the antigen is similar to the blood group. It's called agglutinogen also, other name of the antigen. And they have antibody or agglutinin B, the opposite of the blood group. While blood group B, it has the same antigen B, and antibody A. Blood group AB, both antigens A and B and no antibodies. Blood group O, no antigens, while it has antibody A and antibody B. Clandestine role in blood transfusion, we have to know that blood O is called the universal donor. It donates to all blood groups, while AB is called the universal recipient. The non-self, document 3, page 119. Antigen, maybe it's cellular antigen or it's soluble. Cellular, from its name, it's found in the membrane of a cell or virus. Soluble, it's free, secreted by a cell, and not bound to the cell itself. Antigens, they can be non-self that originate from outside. Example about this: bacterial toxin. Modified self, synthesized by the body cells in exceptional condition. Example about these: cancerous cell. Self is synthesized by the body cells in normal conditions. Lymphoid organs, we have primary and we have secondary. Primary, they are the site of production and maturation of the leukocytes. Mainly, they are the bone marrow and the thymus. While secondary, they are the site of functioning of these leukocytes. They are the spleen and the lymph nodes. In the bone marrow, B cells should recognize the self-antigen. Whenever it recognizes the self-antigen, this leads to its death, while if it didn't recognize the self-antigen, they survive. T cells, they have to recognize the self-HLA in order to survive, while if they don't recognize the self-HLA, they die. Then, these T cells, they have to recognize self-antigen, meaning that this leads to death, while if they don't recognize, this leads to its survival. So it doesn't just means recognize, doesn't recognize, results in. This is a concept map showing the maturation in the thymus and bone marrow. This table showing the differences between the antibody and TCR. Antibody structure, it has 4 polypeptides, 2 heavy and 2 light, has a variable region and a constant region. While TCR has 2 polypeptide, variable and constant region. Localization on B lymphocytes or free in plasma, while TCR is found in the membrane of T lymphocytes. Antigen recognition, cellular or soluble antigen, while TCR, antigen presented on HLA. It's called double recognition process by T cells. Number of antigen binding site antibody, it has 2, while TCR it has only 1.